Welcome everybody to the virtual Watershed Symposium 11 o'clock session today. Your presenter will be Tim Craddock shortly. He'll be speaking on benthic macroinvertebrate identification. I'm pretty excited about this. I've seen the slides and heard his talk, and I learn something every time. So without further ado, let's just talk about the Zoom meeting controls. We're showing that on the left side of your toolbar, you can mute yourself and unmute yourself. Please keep yourself muted while Tim is talking. And then you can start and stop your camera using the little movie camera icon. And if you look for the little call out icon, which looks like a thought bubble, you can ask questions there, and that's the chat window. And I know many of you were with us for the 10 o'clock session, so welcome back. But I think we also have some new people. So if you would like to introduce yourself in the chat window, go ahead and tell if you're just joining us. We'll know who we have. And your speaker will be Tim Craddock. He's with the West Virginia Department of Environmental Protection in the same group I'm in, which is Watershed Improvement Branch. And his area of expertise is the non-point source program. But prior to becoming the manager of that, he used to be the Save Our Streams coordinator. So when I came on board here 16 years ago, first day on the job was a training down at the Isaac Walton League in Beverly, I think. And we did the macroinvertebrates, and I was pretty overwhelmed because I had never been exposed to that before. But as with anything that you learn, when you learn a little bit, it becomes really fun and exciting, and you can't wait to pass that on to others. So that's what Tim's going to do today, and I think you'll find that he can probably answer a lot of your questions, and maybe some of your questions will just be answered by the slides. And I will turn it over to you, Tim.
As Alana said, I'm Tim Craddock. Just a little bit about me before we move on. I'm almost 60 years old next year. And more than half of that time has been spent either employed or being a volunteer for watersheds. So pretty passionate about this effort, and I believe that the passion is something that we have in common throughout this room, and even for many who could not participate in today's session or yesterday's session. One of the wonderful things about West Virginia is we are passionate about our water resources and our natural resources. So it's a feel-good moment for all of us. This presentation will discuss the basic morphological features that aid in field identification. The benthic macroinvertebrates, I'm going to call them BMIs from this point forward. We're also going to touch on the importance of BMIs and how West Virginia DEP's Watershed Assessment Branch uses them. What are BMIs? Everybody take a second. If you know, you can type it into the chat box. It's pretty simple. Benthic part of the word means bottom inhabitant, so they're on the bottom of our streams and rivers. Macro is visible, so we can see them without magnification, but magnification helps, especially with identification. And they're invertebrates, so they don't have a backbone. I'm going to switch gears a little bit and talk about our Watershed Assessment Branch, WAB. WAB is the scientists and natural resource providers within DEP that do the water quality monitoring all throughout the state. They monitor all types of water bodies, streams, rivers, lakes, wetlands. They process the data, they store the data, they analyze the data, and they provide it to the TMDL section. And if you don't know, TMDL is an acronym for Total Maximum Daily Load. And they're the resource providers and professionals that develop, they're called basically pollution budgets for the stream, that tell us how much pollution a stream can get and still maintain a healthy, vibrant ecosystem. I like to call it, instead of pollution budget, the clean water budget instead. So the WAB folks, if you see them out there, give them a thumbs up. They do a lot of good quality work. And then it's been tough this year, as you know, for all of us, but even during these times, they're doing a lot of work. Why do BMIs matter? And why do we collect them? Well, I'll get into that a little bit, but think about that for just a minute. There are many, many reasons. And if you want to type in a few in the chat box, please feel free. This is my Because Pause slide. It's just nice pictures, so take a minute and look at that. This year has been beautiful fall colors. Hopefully you got outside and experienced some of that beauty around you.
Tim, someone named B did tell us that bugs tell us if the water is healthy.
That is correct answer, B. Thank you. One of the primary reasons is they're in our water quality standards. They are the tool, the key measure of interpreting the narrative water quality standards that no significant adverse impact to the chemical, physical, or hydrologic or biological component of aquatic ecosystems shall be allowed. So that's important. So it's the law, it's still there. WAB has some tools to help us evaluate what the biological community, how it's doing. A couple of these tools are family level IBI, and IBI stands for Index of Biological Integrity. We also have a genus level IBI, and coming soon, a fish IBI. All these tools are available. We're still doing this kind of work, but there's some flux that I won't get into today. Perhaps we can talk about that with questions or perhaps another time. The whys are continuing, and here's the science behind the whys. In West Virginia alone, first of all, diversity is key here. In West Virginia alone, the Watershed Assessment Branch has collected 133 different families and 540 different genera of benthic macroinvertebrates. So that's a great deal of diversity, which is critical. Some other reasons: they are the primary food source for all aquatic animals. They're excellent for studies. They represent past water quality, unlike a water sample, which only tells you what's going on presently. They represent this because they've been around for a little while, and they have very low mobility. So whatever impacts them, they tell the story of the impact because they can't swim away. They're abundant in almost all types of water bodies, from the smallest stream to rivers. They're easy to collect with relatively inexpensive equipment and very few people. And some of this equipment can be homemade, so it doesn't have to be anything fancy. And there's been extensive research and great deal of knowledge about life history and stress tolerance. One of the things that I would like to interject here is even though WAB is excellent at doing their job and going out and monitoring our streams and rivers and wetlands and lakes, only a small percentage of those are monitored. That's why volunteer monitoring is critically important to help fill in the gaps. Not only that, so we'll get into the identification part of this a little bit. What we're going to do is we're going to discuss the major groups. We'll target a few features that are beneficial for identification. And I'm going to provide you with additional tips and some from my own experiences and readings. Also, in what you'll see in each slide is a tolerance scale and a size scale. The size scale is in millimeters, and the tolerance is just kind of a range from 1 to 10. And you can pretty much see that low is low numbers and high is high numbers. And that tolerance is how the organism responds to a variety of insults, especially insults at lower dissolved oxygen levels. Just some tips from myself. This is like anything else, like Alana said, you have to learn what you're doing, you have to practice. And that's important. If you get things wrong, that's okay. Just keep at it. It's pretty easy once you get the hang of it. The first tip is to know the general gestalt. Gestalt is a German word that for form and shape. So in other words, if it looks like a crayfish, it is a crayfish. If it looks like a stonefly, it is a stonefly. Once you learn what that form and shape is, you'll be able to identify a wide variety of organisms. Know if the organism is an insect or not. Insects have 3 body parts and 6 legs, but there are exceptions to that, so be aware of those exceptions. When you're identifying a macroinvertebrate, a BMI, try to orient it in a way that's similar to the field guide or key you're using. Most of them are drawn in top view, so once you're looking at something in a small magnifier, orient it in that view and you'll be able to identify it easier. Remember certain movements and shapes. These are usually very consistent across the board. They move a certain way, they act a certain way in the collection pan, and once you identify those, you can easily identify those in the future. So color and size are not good tools to use for this process because color is variable. It's based upon environmental factors, and they could be all different sizes depending upon where they are in their life cycle. So don't use color or size, but oftentimes in identification field guides, those kind of things are referred to. Jump right into the major orders. We'll add something here that most of the animals that you collect doing this are going to be insects, aquatic insects. And aquatic insects have an adult and larval stage. Most of what we're talking about are larval stages. The adult stages are often terrestrial, although there are exceptions to that. And something to keep in mind also, if I tell you something, most likely there's going to be some exception down the road. The Ephemeroptera, or the mayflies, they have 6 legs, 3 tails, but sometimes they have 2 tails, and their gills are attached to the abdomen. So you'll see in the picture the gills, various types of gills. Some of them are fuzzy, some of them look like little scales. Some of them are very hard to see, but they're always going to be on the abdomen. Using tails to identify mayflies, not a good idea, primarily because there are a few families that only have 2 tails. Also, tails tend to fall off during collection. So you could find a mayfly, or you could collect a mayfly that doesn't have any tails at all, and it could still be a mayfly. One of the things you might want to become familiar with this group is how they move. Once you get them in a pan, these animals move around in certain ways, and mayflies have several types of movements. Some are swimmers, some are clingers, and some are burrowers. And the swimmers typically dart around your collection pan really quickly. So most likely, if you see that, even though you might not be able to identify it or catch it even, that's probably going to be a mayfly. Let me go back for a second. Mayflies, as you can see, have mostly low to moderate tolerance, and they're not very big. Most of them are very small, so sometimes they're difficult to see. The burrowing mayflies are typically a little bit larger. Plecoptera are the stoneflies. They are the most sensitive order of all macroinvertebrates. They have low sensitivities to a variety of insults. So if your stream doesn't have stoneflies in it, perhaps there's a problem, but it also could be a stream type because stoneflies typically like fast, clear, cold water. They're more common in headwater streams or fast moving streams. So if you have a slower moving stream, you're most likely to not find stoneflies. So that doesn't necessarily mean your stream's not healthy. It's just not an environment they prefer. Stoneflies have 2 tails. They have gills, but they don't have gills on the abdomen. And oftentimes their gills are difficult to see. If you do see them, they're going to be up around the head on the legs. So sometimes the legs are a little bit fuzzy. They have 2 claws on the end of their legs. They're very adept at staying in fast water. They can hold onto rocks very well. Sometimes they're hard to knock off the rocks. A little tidbit about stoneflies once you catch them. Again, they'll move around real slowly in your pan, and they'll do something that looks like a push-up. And so the body will move their body up and down. And what's happening is they're trying to move water across their body and across their gills to obtain oxygen, because that oxygen in that pan may be lowering a little bit. So they tend to do push-ups to help them breathe. The next group, order Trichoptera. These are the caddisflies. They basically look like a small grub or caterpillar. Their body is soft, their head is hard, their legs are located up near the head in about the third of the thorax. What's interesting about them, and really their own house. They build retreats, they build cases of a variety of shapes and sizes. And sometimes these cases can be used as an identification tool, but I would caution you there, because they're also going to use whatever's available to them. So just a case looks a certain way, that doesn't necessarily mean it's that particular family of caddisflies. There's also a group called the net spinners. They do not build cases. They look similar to one another, but they build what's called retreats or nets. And nets are used to hide themselves, but primarily they're used to catch food. And there's one caddisfly that's the exception. It's the green sedge, as a trout fisherman would call them. They do not build a case. They do not spin a net. They're the only true predatory caddisfly. However, all caddisflies have the ability to spin silk. They're closely related to butterflies and moths, so that's where that relationship comes from. And it makes them very successful in the environment. They can live in a lot of different kinds of waters. And as you can see from the scale above, they're not real big either, and their tolerance range is from very low to moderate. In general, these first three are the most sensitive groups that you'll collect in the environment. A few pictures. I'll give you a few minutes with that slide. As you can see, they all basically look very similar to one another. I will tell you something about the movement here. Once they're in the pan, once you've collected them, they tend to look like they're doing push-ups or form themselves into letter C's. So if you see something that looks like it's actually doing a little bit of not push-ups, I'm sorry, crunches. If you see something that looks like it's doing a sit-up or a crunch, it's most likely a caddisfly as well. Odonata is the dragonflies and damselflies. These BMIs have been around for a very, very long time. They're well adapted to their environment. They're more typical of wetlands and ponds and lakes, but they're sometimes collected in fast-moving streams as well. They're very robust. They have big eyes. The dragonfly's body is thicker and rounder, and the damselfly body is more thin. Their gills are not visible, although, and they don't have tails. Now, looking at that one picture, especially the picture for the damselfly, you would say, Tim, you're lying. There's tails right there. I see them. Well, those are not actually tails. They're gills, although they do have the appearance of tails. One of the coolest things about a dragonfly is their lower lip or labium, which shoots out of the mouth if their prey gets too close. It's one of the fastest things in the natural world, and it's very effective at catching their food, and sometimes their food is larger than them, and they're able to deal with that. So they're very strong and powerful organisms. Some have low tolerance, but most of them are moderate to high tolerance. And their size, as you can see, is a little bit bigger than the previous groups we talked about. Coleoptera is the beetles. This is the largest order of insects, period. There's more beetles than anything else on the planet, but most of the beetles are not aquatic. So few of them are aquatic, most of them are terrestrial. So when you collect BMIs, your number of beetles will probably be lower than most other things, but there are some common ones you might collect. Primarily the water penny and the riffle beetle. What's interesting about the beetle also is some of them, the adult stages are also aquatic. So the larval and adult stages are both aquatic, unlike most of the other BMIs. And I have a picture there of what I call the pterodactylid beetle. And I just like that word because pterodactylid is a cool word. It looks a lot like riffle beetle. So if you ever collect one of those, be careful with your ID. You might have something a little different. They have a low to moderate to even a high tolerance. So they can live in a variety of environments. And they also get pretty big. Megaloptera. These are the alderflies, fish flies, and hellgrammites. There's only 2 families in this order. Their distinguishing features are the many filaments that are along the abdomen. The filament looks kind of like a leg, but it's not jointed, so that's how you can tell the difference. The fish fly and the hellgrammite has 8 filaments. The alderfly has 7 filaments. Also, the fish fly and the hellgrammite have a forked tail, whereas the alderfly has a single tapered tail. Excuse me. These are one of the largest invertebrates you're going to collect, and people who are fisher persons like to use the hellgrammite and fish fly, especially for smallmouth bass fishing. So excellent fishing bait. And they are pretty big also. The adult version of these are even cooler looking. Their wings are huge, their claws are huge, but they can't bite. They just kind of climb out of the water and sit there until they perish, which is not very long. Diptera, these are also insects, and they can be confusing because they do not look like insects. Their larval forms do not have any legs. They may, however, have leg-like features. So if you look at the green one, it looks kind of like a caterpillar. There's sort of things like legs sticking up there. The other one on the left, the red, and the other whitish color one, they also look like they have legs, but those are prolegs located near the head and the tail. So basically, be aware of Diptera because they're insects, but they don't have legs. They do have other identifying features that can be useful in their correct identification. Their tolerance is all over the map, from very low to very high. Many different varieties of Diptera can live in organic and polluted streams, so they tell the story of those kinds of streams. Just a few of them that we commonly collect: non-biting midges, chironomids. They're difficult to identify, primarily because they're very, very small and hard to see. So in your collection pan, if you see a tiny little thing that looks almost like a thread that doesn't have any legs on it, most likely that's going to be a chironomid. And especially if that thread is red, that's definitely a chironomid. Also, that their movement is very erratic and they tend to wriggle around a lot. So you can tell them that way as well. Another one commonly collected is the crane fly, the tupulids. Much larger, pretty easily identified. You can see their segments clearly. They have hairs along their abdomen, and they have a really weird tail. Sometimes it looks like a balloon on the back of their body. Can't see a head. Their head is not distinguished. I'll take a minute and tell. I think this story is funny. It might not be funny to you, but it's funny to me. I used to have a German Shepherd I played with a lot, played frisbee with her when we got home from work, and took her for walks and all that. And one day I was playing with her, and this big red squirrel came across her yard and started climbing a tree and then fell off the tree. So I run over to see what's wrong with the squirrel. In the meantime, I'm trying to keep the dog away. And the squirrel, and I'm not watching where my hands are, so the squirrel apparently was just knocked out a little bit, and it woke up and bit my hand, tore my hand to pieces. Ended up, blood all over the place. It was messy. And unfortunately that day, I did not have a vehicle, so I had to wait for my wife to get home. And in the meantime, I had wrapped my hand up, and the squirrel just kind of laid there. So I put the squirrel in a box, and I thought the squirrel was dead. Eventually it did die. But anyway, that evening, really late in the evening, I ended up in the emergency room, and this doctor was stitching up my hand, and I ended up with 12 stitches from the squirrel. But there was this insect in the ER, and it looked like a giant mosquito. If People call them mosquito hawks or gallinippers or lots of different names. And the surgeon, or the surgeon started freaking out and just said, giant mosquito, giant mosquito, giant mosquito, and started throwing things at this insect. And I tried to explain to the doctor that that wasn't a mosquito at all. That was a pulid, and it can't bite you. It's not a mosquito. It didn't have any mouth parts. So sometimes the adults provide you with interesting opportunities to teach. And that was definitely an interesting opportunity to teach. Another one that's commonly collected in the dipterans, it's a black fly. Look like a little bowling pin. Their head is the body is wide, the head is small. They have hooks on their abdomen that allows them to maintain themselves in very fast water. And they also use silk, similar to the caddisflies, which I didn't know. And one time I saw one actually come loose from a rock, and it reeled itself in on its silk back to its point of origin, which I thought was pretty cool. A few pictures. And as you can see, the illustrations look very similar to the pictures, but the size and shape and form, the gestalt of the Diptera are kind of all over the place. Now we're getting into things that aren't insects. So things that aren't insects have usually the easiest way to tell something if something's not an insect is it doesn't have any legs, with the Diptera exception, or it has more than 6 legs. So these are the crustaceans. These are the crayfish, which Eddie is familiar with. And if you have not collected a crayfish, then you might not want to, you don't know anything about a creek, basically. No, I'm just kidding. Most people, as their kids, always collected crayfish. They look like a little lobster. They have 10 legs, and they have little flipper-like things on the end of their abdomen, and a couple of their legs often had large claws on them. The scuds and the side swimmers, they look like a little shrimp. They have a few more legs than the crayfish, and they tend to have a sideways appearance, which is why they're commonly called side swimmers. Really quick movers in the pan, and typically they're not going to be found in riffles as much as other ones. They'll be more related to vegetation and rotting logs and leaves and things like that. The other crustacean is the aquatic sow-bug, also with 7 legs. Most of you are probably familiar with the pill-bug. Well, this is the aquatic cousin of the pill-bug. It tends to like the same kind of environment, like organic areas of leaves and rotting logs and things like that. Crustaceans generally have a moderate to high tolerance, so they can live in a wide variety of environments. And as you can see, crayfish get really large, and scuds and sow-bugs, not so much. A few pictures. And there's it's hard for me to see because there's some blockage here on my screen. But if you write down that website that's right under the crayfish, I would suggest you go to that and just kind of learn some things about crayfish. One of the unique things about West Virginia is we have some indigenous crayfish. It's very specific to various watersheds throughout the state. And there are efforts in some of those areas to protect that indigenous species because they are rare or endangered. A couple of examples, there's an Elk River crayfish, there's a Greenbrier River cave crayfish, and there's one specific to the Guyandot River as well. So there's volunteer opportunities, there's studies for those specific animals, and maybe something going on in your area, so it might be worth checking out. The annelids are the worms and the leeches, and they look like their gestalt is worm. If you see a worm, it's going to be an annelid of some sort. Aquatic worms look exactly like earthworms. They have many segments. There are a variety of oligochaetes, though, that could have different number of segments, and they are smaller or they're white colored or whatever, but we don't differentiate between number of families in aquatic worms because they all have the same tolerance. And it's really difficult to differentiate family to the family level. Leeches are also segmented. They have specifically 34 segments, but don't bother to count because you'll never be able to do that. They have suckers on the ends of their body, sometimes on the tail and head, and sometimes only on the head. And the flatworms are included in this group also, the turbellarians. I only included them here because they're similar in appearance, but they're not actually an annelid. Turbellarians look like they're very small typically, and they often look like leeches. So be very careful when you're trying to identify a leech versus a flatworm. The difference is sometimes difficult to see, but the flatworm won't have any segments, and the eye spots look like they have cross-sided eyes. And as you can see, their tolerance is high or very high, and leeches get very big, and so do worms. Flatworms are very small. There are exceptions to that. There are families of flatworms that are very intolerant to toxics. So again, always going to be an exception. So when I'm talking here, I'm talking in generalities. A few pictures. And this is what the pictures at the bottom are what I mean when I say that the aquatic worms look a little different from one another sometimes, but they're still aquatic worms. When it's the mollusk, mollusks are extremely diverse, the snails and the mussels and the clams. And the North American rivers and streams have some of the most diverse populations of mollusks on the entire planet, which is pretty neat. And these animals have been studied a while. They're very critical, especially in large river environments, to the health of those environments. So a clam or a mussel, they have two hinge shells. And primarily you can tell the difference by the way they look. Clams are more rounded than mussels typically, but again, there are exceptions to that. There are some invasive species of both mussels and clams that we're concerned about. The zebra mussel and the Asian clam are two examples of those, and they've been carried on by into other watersheds by barges and boats and things like that. And unfortunately, they've been carried into some of these areas by fishermen, because what happens is their larval form will attach to the boat and ride along for a while, and you go to the next lake or river or creek, and you end up introducing something. So if you are a fisher person and you have waders or you have a boat, make sure you clean your waders really well. Make sure you clean your boat really well, because it could be that you're introducing something that you didn't mean to introduce. Their tolerances are kind of all over the map, from low to moderate to high. Most of the time, they're going to be low to moderate, especially the mussels, and the clams are going to be moderate to high. And they get pretty big, as you can see in that picture, and you see from my scale below. Snails are pretty easy to identify, only one shell. What we used to use to identify snails is a left or a right. So if you hold a snail up and you face point toward you, it's going to be, the opening is going to be on your left or your right. And we divided this grouping into categories based upon that orientation. Well, come to find out that orientation doesn't really work very well, and it's not consistent. So it's better to identify them in a different manner, especially if you're going to categorize them, and that is using operculate or non-operculate snails. And the operculum is a door. And if you have a snail shell, especially a live one, and it's enclosed in a door, then you have an operculate snail. If it doesn't have a door, it's a non-operculate snail. The other difference is the operculate snails actually have gills, and that's how they breathe. They can stay underwater for a while. And typically they're a lot less tolerant to insults. So they're on the low to moderate end. The non-operculate actually have a lung-like structure that allows them to breathe underwater, and they can actually take gulps of air from the surface as well and breathe. So they can tolerate a little bit more pollution, a little bit more organic input. That's one way to tell the difference. And you can see a few pictures on your right. There's also a snail that looks a lot like a clam. So be careful. We're going to switch gears a little bit, and we're going to talk about how we evaluate these organisms once we collect them. And I'm going to go through just an example here of a data sheet from the Save Our Streams program. This is the level 1 evaluation. And there's data sheet that once you collect all your animals, you start recording. And at this level, you don't necessarily count everything. You estimate their abundance. You can see the scale below. If you see something where you have greater than 50 of those, most likely you can tell if there's greater than 50. If they're common, they're between 5 and 50. If they're rare, they're less than 50. So you record these numbers in all these categories. Something to be aware of when you begin your identification is some groups, like stoneflies, mayflies, caddisflies, and some others, have the possibility of finding many different varieties. We call them kinds, also families specifically, are more common in that group. So the data sheet sort of tells the story also. The data sheet will tell you, well, there's a little, you see the little gray box. That tells you that the possibility of having another family or two is there. So it's something to look for when you're doing your collection. So this is a typical example. You'll see that the stonefly is a C, the mayfly is a C, the dragonfly is an R, so forth and so on. And that there's a website down there at the bottom of the page that you can go to if you want to learn more about this scoring possibility. We also have, the program also has a variety of spreadsheet tools. But all you have to do in that situation is enter the numbers, and the spreadsheet does the work for you. Here's the metric calculations, and the metric calculations are what we use to evaluate, to get a quantitative evaluation of these organisms. These metrics are based upon what the Watershed Assessment Branch uses. If you'll recall, the genus level IBI and the family level IBI. So they're similar to that, maybe not as difficult to calculate, or maybe not as many in some cases, but they're very similar. And once we get a metric calculation for each one, we combine these metrics into an overall stream score, and you can see how that is done. So you'll take, if something's abundant, that converts into a 6. If something's common, that converts into a 3. If something rare, that converts into a 1. So we write all those numbers down on our sheet here and multiply the tolerance value, which gives you a tolerance score. We record the number of different kinds, and we sum all that information, and we come up with 3 different metrics. Total taxa, which is the total number of kinds. EPT taxa, which is the number of kinds of mayflies, stoneflies, and caddisflies, and an overall biotic index, which is kind of the overall tolerance of your entire collection. So the lower that number, the less tolerant your organisms are. And you can see in this example, this kind of was the middle of the road. It's pretty good. It's not optimal, but it's still pretty good. Basically, what you want your stream to be is optimal or suboptimal. If you find that your stream is frequently in the marginal or poor categories, then there's a problem. And we need to investigate reasons why that's a problem. Talk a little bit about resources. I really encourage you to visit the Save Our Streams website if you haven't. There are a ton of resources there on macroinvertebrate identification and other ecological issues that volunteers can deal with. What's interesting and cool about these animals is that once you learn how to identify them and you have a preferred key preference, and it doesn't have to be Save Our Streams, although I think I'm a little biased. I think Save Our Streams keys are some of the better ones. But these animals are ubiquitous. They're worldwide. They occur all over the planet, and they're very similar all over the planet. So you can take a key from West Virginia or Michigan or Minnesota or Georgia or wherever and use that same key to identify these animals wherever you are. So they're really interesting, and they are the canaries in the coal mine, so to speak, for many, many types of aquatic environments. So whatever your preferred resources are, learn that and get good with that particular guide. But on the West Virginia Save Our Streams site, there's a lot to choose from. If you recall from when we started this thing, we talked about an activity, and this is not something that we're going to do together here. We're going to do this sort of on your own time, but I'll go through what this activity is. Again, the website there is how you get there, but you can probably do a search and find it also. And If you can't, just send me an email and I'll send you to the link and you can do the activity. Those of you who want to participate, we have a week. So today is the 6th. You have until the 13th to finish this. And the purpose is to identify 30 different BMIs. And I want you to identify these organisms to the lowest taxonomic level possible. Be as correct as possible because it's going to matter when we evaluate all our entries. Because I was able to get some really cool prizes. They are related to benthic collection or analysis or something. We use a company called BioQuip for buying nets and such, and have for many, many years. And actually, BioQuip helped us design the net that is used for the Save Our Streams program. So it's a very cool company to work with. They're out of California. I actually had a chance to visit with them once on a water conference. It was out in San Jose one year. That was really good people. So there's some, all you have to do is follow the instructions. There's a form to download. You can download the Word version or the PDF version, complete it, fill it out, save it to your hard drive, save it to your computer, save it to your phone, and email it to me, and I will do the evaluation and we'll figure out who the winner is. Now, at the if there is a tie, then I'll come up with something to break that tie. And right now, I'm not sure what that something is, but we'll figure that out. But again, I just identify these animals to their lowest taxonomic level and be as complete as possible. So my preference, hint, 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 is to use the taxonomic name, not the common name. That might help you gain a few points here and there. And you can use whatever tools you want to. You don't have to do this by memory. There's an example right here of the Save Our Streams 2-page BMI guide, but there are many, many others that you can use. So feel free to use what you want to. Just some acknowledgements before we go to questions or Jennifer Gillies and the Capon Institute. They're the organization who had worked with me many, many years ago and developed these images for the guides and keys we use today. And we continue to use them, and I get all kinds of comments even today for other organizations wanting to use these images in their publications and their guides and keys. So they're widely used all over the place. So if you see them elsewhere other than Save Our Streams, you know where they came from. Also want to acknowledge the contribution of the Watershed Assessment Branch. They do wonderful work and they've provided some information for this slideshow. Alana helped me sort of go through this process and sort of get my head around it a little bit. But most of all, I want to thank all the Save Our Streams volunteer monitors and all the watershed groups out there doing work. I implore you to continue to do that work. It's extremely important. It's extremely valuable. And we listen. Believe it or not, it might not sound like it or feel like it some days, but we truly do listen. So I thank you for your time, and we'll take questions.
All right, thanks, Tim. The only question I saw in the chat that I can recall actually being a question is whether you were going to talk about the development of the IBI.
Answer is no, because it's it would be a very detailed and lengthy discussion. But just to give you a background, I mean, the IBI was developed by a collaboration, kind of like what Dr. Henry Stone was talking about in collaboration. DEP and EPA and many others collaborated on the development of family level and the genus level IBI. And it involves sampling hundreds of different sites, looking at hundreds of different organisms, and coming up with a sense of what these organisms are telling us from site to site. So that's the crypt note version, but it's more detailed. Now, if you want more details, certainly email me and we can discuss those, and I can send you the documents associated with those IBIs as well.
All right, we just had another question come in from Harry Stone. What role do volunteer monitors play in West Virginia? Is there coordination with other states doing similar things?
It's a good question. Yes, there is. Especially in the Bay region, there are a lot of organizations doing volunteer work. The Chesapeake Monitoring Cooperative, the Isaac Walton League, some of these have been around a long time. The Save Our Streams program has been around a long time also. But, one of the things that's I think interesting and cool about this, you don't necessarily have to use a standard operating procedure from one organization to another. A lot of these collection methods, a lot of these tools are the same across the board. So there is certainly opportunity to collaborate, and there is currently collaboration also. Just recently, I had a request from the Chesapeake Monitoring Cooperative and the Isaac Walton League to upload all our data from the volunteer assessment database to their data set. So that's something we're working on. We found a lot of hiccups doing that process, but we're getting there. So the answer to your question is yes, absolutely, there's a collaboration. And in terms of the importance of volunteer monitors, I'm biased because these people excite me, this work excites me, but they are critical because they are the feet on the ground. They are the local eyes and ears of their waters. And oftentimes they are in areas where there hasn't been any sampling or there hasn't been any collection. So that's one other thing that you can do if you have that situation, is if you can submit data. And we do accept volunteer data to a degree. The identification part and the benthic macroinvertebrate part is a little bit more difficult. And even our own watershed assessment branch sends these BMIs out to a lab to be identified. So it's not necessarily something, field identification is not something that we accept necessarily, but they are still important regardless, because they do tell us trends, and you can say something's wrong here, come out and help me learn about my watershed and figure out what's happening. So hopefully I answered the question.
All right, and just one more question came in from Callie Sams. She wanted to know if there's seasonal variation in what you find.
Yeah, a little bit.
I've memorized the question. I hope I got it right.
There are. One of the things is that when the first metrics were developed, the West Virginia stream condition insects, they were primarily stream and fall collections. So what we learned was based upon stream and fall and summer collections. And also too, these animals grow and develop at different times. So there's definitely variations, but they're there in the wintertime as well. So even in the wintertime, you can go out and find a healthy population of invertebrates. In the early spring and winter, oftentimes it's more difficult because the weather is nasty, it's cold, it's harder. But also they tend to be smaller because the adults or the larval stages haven't really had a chance to grow up much. So the population tends to be a little smaller. So identification therefore is more difficult. So seasonal variation, we're learning over time, is less and less of a problem, or less and less of something you need to pay attention to. You can go out almost any time of the year and find out the biological condition of your stream.
All right, thanks, Tim, and everyone for your great questions. I did also get a comment that the bit.ly link wasn't working. It might be your browser, but it is working for some people, so we know we have it right. I'm willing to send the Word document with the quiz on it to people, but I guess you actually need the link because that's where you'll find the images. So
yeah,
we'll work on that.
Just have people email me, and if they're having problems with the link, I will guide them to the right place.
Oh, okay, thank you. So we put your email in the chat here. Maybe can you do that for us, Tim?
Yeah.
Okay, so Tim's gonna put his email in the chat. Wonderful. Okay, thank you.